Ever wonder why a virus hits one cell but leaves its identical twin next door totally fine?
It's a huge puzzle in biology, so let's play detective and see what's going on.
So it starts here, some cells get infected, and some just don't. It's an unequal fight. The biggest problem? The virus totally wipes the crime scene. It changes the cell, hiding all the clues.
So to solve this, scientists needed a way to look back in time, before the infection ever happened. And that's where this rewind method comes in. It's basically a cellular time machine. Pretty cool, right? Here's how it works. They barcode cells, create twins, infect one group, then match the barcodes back. Simple. So, what did this time machine reveal? Well, for SARS-CoV-2, they found a prime suspect.
It turns out the virus isn't random at all. It actually hunts for cells in a specific TIG1 high state.
And get this, TIG1 high isn't a different type of cell. It's just a temporary state it's in.
And here's the proof. You knock out those TIG1-related genes, and boom, the infection rate plummets.
Okay, but what about another virus? Does the flu have the same type? Nope. Influenza goes for a totally different target, a KRT8 high state. It has its own preference. This is all great in a lab dish, but does it actually matter in our bodies? Let's go to the lungs.
And yep, that TG1 high signature is right there in our lungs, in the exact cells SARS-CoV-2 attacks.
So what does all this actually mean for us? Well, it's a pretty big deal for understanding risk.
For example, people with lung fibrosis have way more of these super susceptible TIG1 high cells.
And it's not just disease. Things like smoking can actually increase the number of these vulnerable cells.
So the big picture is, it's not just about the right door. The cell's internal state is absolutely crucial. Which leads to a fascinating thought. Could we one day predict our viral risk just by reading our cells?